We've got Laurel Robertson. She's going to talk with us about the March of the Microbes, or March of the Microorganisms as it was on this sheet. So Laurel, thank you, and here's your microphone.
So, hi, I'm Laurel, and I'm really excited to be here with you today. I'm a pretty recent convert to the whole story of soil food web organisms. And like any convert, I've got that kind of enthusiasm. But first, I got to be honest with you. I'm going to quote Stephen Stills at Woodstock. Man, this is my second public performance and I'm scared shitless. But I'm not sheetless. I got my notes. I've got slides, so I think we're going to be okay. Yeah. Okay, to begin with, I'd like to ask everybody to close your eyes and imagine a plant. Let's say a tree. Just imagine your favorite tree. You don't have to close your eyes. Get it in your head and I'm thinking that most people see something like this. In fact, when I Googled tree, that's what came up. But a lot of you here, of you plant people, are smart enough to know that that's not the whole story. This is actually, whoops, sorry, I'll get used to it. This is what a tree really looks like. And this is what we've lived for decades or for centuries now. We have, as humans, an above ground bias about what we can see and ignore the rest. The bottom part of that, which is called the rhizosphere now, is as much a part of the tree as the top part. In fact, I'm going to argue in some ways it's even more important. The rhizos is the Greek word for root and sphere, so it's the area of roots. And what all goes on down there is an amazing story that's really just come to light in the last, I'd say, 3 decades, and it's revolutionary. Okay, so let's start early. When a seed first sprouts, the first thing the plant does is send down a root. And it waits until that root is well established before it gambles to send a shoot or a leaf above ground. That's how important roots are. Only after that solid foundation of root is established will it begin to send shoots and leaves up to gather sunlight. And to me, that's meaningful. In fact, it's kind of metaphysical because as was expressed in the shortest poem I know, written by a good friend and a great poet, there's a lot going on within a little thing. So an important concept in environmental science is that of the good old food chain, which I'm sure everybody studied and we're familiar with. There's your primary producers. There's a producer, primary consumers, eaten by secondary consumers, eaten by the tertiary consumers, and on in the chain, right? Well, this is also a little bit of an above ground bias because nature doesn't end at the soil line. In fact, what's going on underneath is, of course, as dynamic and as complex and as beautiful as anything going on above ground. It's just that we can't see it. It's in the dark. So here is a model of what's going on underground, known as a food web. It's got primary producers. It's got the primary producer. It's got primary consumers, secondary consumers, and so on. But it's not as linear a process underground as it seems to be above ground. Many of the actors in this kind of newly discovered soil food web play many different roles. Sometimes they're primary consumers, sometimes they're tertiary consumers. So it can be a more complex and a more dynamic. And they call it a soil food web because it's like a whole mishmash of activity going on underground, which we are really, as science, just beginning to understand. Okay, so why are we just getting to this? Why have we studied the above ground part so much and are just getting to the below ground? In a big part because it's hard to see. Not only is it dark down there, but all the organisms that live down there have no reason to have color or melanin. They're basically, even under a microscope, invisible. They're clear, they're transparent. So even when scientists were looking in a microscope, they're hard to see. And so what's hard to see, we kind of, we want to look at what's easier to see. We want to study what we'll study and what we can see. So until the middle of the last century, so the 1950s, say, they didn't have a microscope that could see these critters underground, these transparent things. But with the invention of a shadowing microscope, they began to be able to see them. Okay, a regular compound microscope has a light that shines up straight from the bottom. And under that, you can't see transparent things very well. But a shadowing microscope takes that light and makes it come in at an oblique angle so that all the organisms throw shadows. And now there's definition to them and you can see them. So it has made a big difference in science, just the technology of it. And so about the time that happened, well, you'd think, oh boy, soil biology is really going to take off, but that's not actually what happened because this is the post-World War II era and chemistry became the darling of, chemicals became the darling of all kinds of science. And they studied the chemistry of soils and plant interaction more. And that reached its zenith with the Green Revolution following World War II. And basically it explained everything with a plant as a chemical interaction and looked at the soil as basically just a vehicle to hold the plants upright and maybe supply NPK, which is nitrogen, potassium, or nitrogen, phosphorus, and potassium, the three main nutrients that at the time they thought that's all plants need. But now then, since the, say, mid-1980s, biologists armed with shadowing microscopes and other things began to see that there's a lot going on underground in a symbiotic relationship between plants and all these different microorganisms. And so in the mid-1990s, Elaine Ingham and a team at Oregon State University mapped out the original soil food web, which this is not their drawing, but similar pattern, showing the interactions, showing the symbiotic relationships, and also the, who consumed who in that. And the world of soil microbiology has only exploded since then. The things they're finding out it's hard to keep up with. You can't, I have trouble staying abreast of even just the basic things. Well, so who am I and why are you listening to me talk about all this? Fair question. And full disclosure, I am not by training a scientist or a biologist. My degree's in history and anthropology. And, but I was always interested in plants. I studied HRM, permaculture. I became a master gardener and a master naturalist. I've always been interested in the natural world. And then when, as an historian, I got interested in the book, Dirt by Montgomery, I was amazed and maybe disheartened to read how every civilization that we can track has, in a big part, failed or fallen because of the way they treated their soil, because of what happened. They played out their soils. And I started looking at soil biology as hopefully that we will not befall that fate, that our civilization will be one that can figure it out and get and live with the soil instead of mining it. Coincidentally, about the time that was coming out, the world of microbiology had really just broken that soil web, food web code. And now this, after only, what, 12,000 years of practicing agriculture. So, wow. Um, and to be clear, we are not the only culture that has ever learned to live sustainably with the land and with agriculture. In fact, we haven't done it yet, but there's some glimmerings of beginnings. But finally, with soil food web, we're able to understand in terms that mean something to us and our mindset, what's going on. So my entry into this world, after I read the histories of it, was through Elaine Ingham's Soil Food Web School, which is a very rigorous multi-year study course online that gave me enough basic understanding of botany and soil microbiology for me to be able to look at soil under a microscope and tell what's going on in there, or tell who's in there, and from that understand how it's interacting, what's going on, what the changes are. I want to, full disclosure, I am not a representative of the Soil Food Web School. I do not get any profits from it. I'm just recommending it as a credible source of information if you're interested. So the background knowledge I got from there has enabled me to understand some parts of this fire hose of information that is becoming available to citizen scientists around the world, which I consider myself a citizen scientist. And there are so many like-minded people who are doing all kinds of wonderful research and experiment and just explanation of what they're finding. And so it's very much a web of resources out there, underground and above ground. And you can jump in and find out more about it too, because there's all kinds of good websites, and I have some books I can recommend. Okay, so that's enough about me. So what about you? Now, how is this going to be of any use to you in your everyday practice with native plants, say? Well, what if you're trying to grow plants in soil or not soil like this, which I think a lot of us are? Or what if you're trying to recover from a fire or a flood or even new house construction? These are soil killing events. And, or maybe you just want to get rid of your, carpet grass and put in a native plant, a native wildflower meadow. Or even more ambitious, maybe you want to grow a madrone tree. Understanding and promoting the right balance of microbes in your soil can help make all these goals happen more quickly, happen faster, and more successfully. Okay, so here's a standard biological description of how land recovers and plants recover after a devastating event. See the fire over on the left-hand side? But it could be any devastating event, even house construction. In fact, house construction is in some way worse. First to grow are the annuals. See where it says 1 to 2 years at the bottom? Weeds take over. And that's a good thing, because bare soil is the worst thing you can have. So annual weeds, usually they have a lot of stickers, usually they have a lot of defenses, because these are pioneer plants. And they're there to, it's kind of like being a Martian or staking out a living on the moon. This is hard, and what they're doing is pretty heroic. Okay, and so after the first year or two, then among those weedy plants, grasses and perennials, short perennials, maybe shrubby perennials will begin to grow. And then after they have shaded the soil, added to the organic matter, in many years, look at the 5 to 150 years, grasses, shrubs, and well, this is, we don't have hickories, but you get the idea. Larger trees. So this is a succession, and now we're into intermediate species. And finally, the climax growth after many, many decades will be hardwood trees, bigger hardwood trees, and pines too, and conifers. This can take up to, look, 150 years. I don't have that kind of time. And I've got 20 years tops, so I've got to hurry this along. So how can we make this pattern? We need to follow this pattern. You can't shortcut it, but you can leapfrog it and make it go faster. What's not being shown in this is, guess what? What's going on underground? Because as the species are changing above ground, so are the entire population of species below ground. And which comes first? It's kind of a chicken and an egg thing. Are the microbes creating the new plants, or are the new plants creating this new population of microbes? I think it's both. And this is nature. It's not either, it's not one or the other, it's both, and they feed each other. Okay, so how does that work? Let's start at the beginning. Photosynthesis, which is, in my book, the most miraculous thing that goes on the planet. It continues to just blow me away. It is the source of all life on Earth. I think I can say that as a blanket statement, and there's not many blanket statements you can make in science. And plants are the creators of most of that. They're not the only thing that photosynthesizes. There's a few. In fact, there's even some bacteria at the bottom of the deep sea trenches that can photosynthesize without the sun, but that's such an exception. For the most part, it's plants. Okay, and they take up air, water, and with the power of sunlight, they create sugar. Sugar is the gold standard for every living thing. It's the beginning of energy. It's portable sunlight. And I'm so blown away by this that I want to get a tattoo of photosynthesis, and I can't quite figure out how it's going to look. So if any of you have some really good ideas, come see me after, please. Okay. Okay, so now here's a typical drawing. In fact, I think it's made for fourth graders, showing how plants photosynthesize and gather up nutrients. And it is simplistic because you see light energy, carbon dioxide, water, we let off oxygen, and then glucose, which is sugar. And then the roots take in water and minerals. Okay, it's nice, but that's not the real story, because that glucose going off into the air, and I think what they mean that it is going to be dead organic matter, that glucose is way too valuable for a plant just to discard. That is the most sought after thing in the environment. It's universal currency in the natural world. Photosynthesis is the only way to get it, and everything needs it. And so plants not just going to give away that photosynthesis, it's going to spend it wisely, and we'll get into that. And the roots are just sucking up minerals because they're just all available there. And in water, no problem. Yeah, plants they're just in that one little space. I mean, they've got, let's see, to quote the genie in Aladdin, phenomenal cosmic photosynthesis powers, but little bitty living space. They can't go anywhere. So they can't go get more minerals, more water. Once they've exhausted what's right there in their little part of the rhizosphere, that's it. So what are they going to do? Well, millions of years ago, they worked out an elegant solution. They hire other microbes to go get it for them. Specifically bacteria and fungus are their partners underground for getting more of that. And it's a busy world under there. It's a bustling marketplace underground, and just seeing parts of it under a microscope is really fascinating. I know I'm not going to proselytize. Okay. Okay. So here's a more realistic drawing of how plants and nutrients get plants get nutrients and take part in this underground network of the soil food web. And even this design needs a little tweaking because it shows just the tree, the plant, creating organic matter, which is then consumed by the fungi and the bacteria. And actually what's going on is that, did it change? Yeah. Sometimes the plant is directly feeding the fungi and the bacteria exudates, that is sugar in liquid form, through its roots. And in fact, plants spend up to 50% of all of the sugars they produce exchanging them with underground microbes. And that's a big investment, but the payoff is good for them too. Yes, so those secondary, okay, so the primary consumers get this sugar from the plants, and then they do this trade. They have bacteria and fungus make acids, which can dissolve minerals, which can dissolve rocks in the soil, which can dissolve nutrients out of the soil substrate. Plants have no acids. In fact, acids kill plants. Plants can't deal with acids, but they sure can get them from the bacteria and the fungus who have dissolved them. We'll talk a little bit more about that later, how they do that. But then so here's the bacteria and the fungus full of sugars from the plant. We're all having a great living, and here come predators, what's my name? Here come the predators. Nope. Okay, it'll stay on there. And the predators, which are protozoas, nematodes, and some other things, I'll show you details about them, consume the bacteria and the fungus, and particularly bacteria are juicy little things. They have a really high nitrogen component. They are 5 parts carbon and 1 part nitrogen. Almost every other living thing, including us, is 30 parts carbon and 1 part nitrogen. So to get enough carbon, they've got these other earthworms and protozoa have to eat a lot of bacteria, but now they've got way too much nitrogen. So they poop it out into the soil. And then it's in a plant available form too. And nitrogen is liquid gold to all the soil food web. And it does not sit there long. It is snapped up by a plant, if a plant's available, or some other organism. But nitrogen doesn't just sit there in the soil and get washed away in the first rain. It is locked up. It's always there, and it is not going anywhere, but it is being exchanged. But it's not going to go down to the Gulf of Mexico and cause algae blooms. So a great way to keep soil nutrients in the soil is to have them locked up in these organisms, which are always trading and moving around. So everybody's got a chance to get some. So this is a poop loop, if you will. You know, what goes around comes around there. Okay, and then here is more detailed explanation of the interactions between the plant roots and the fungal and the fungi and the bacteria. Plants can be very specific. I'm not going to use the word intelligence because we're not saying that about plants, but their ability to control their environment is amazing. They can exude just the right kind of sugar or the right chemical that will encourage that fungi or bacteria to go get exactly the nutrient that plant needs. And that's still being studied, and it's way above my pay grade to understand how all that works, but it's, there's evidence that it's really specific and it works. Yeah, it's a complex and finely tuned system. So this is one of the reasons why we need to have plants in the soil at all times. If there's not a plant there photosynthesizing and sending sugars down into the rhizosphere to feed the microorganisms, which are then trading it all around and bringing things that the plant needs. If you break this cycle and have no plants, then the microorganisms are going to consume themselves, and then you won't have them when you're going to plant. So letting fields lie fallow or areas of soil sit without plants is always a mistake. It is, what does Jesse Frost say? If the soil is not consuming plants, it's consuming itself. So really important to keep plants in the soil. Okay, and beyond the primary consumers are the secondary consumers. I got that. Oh, sorry, I'm going backwards. Yeah, I'm new at this. Okay, I'll get there. We got it, I think. Yeah, yeah. All right. Okay. Okay, so bacteria. Let's go with these primary consumers. Bacteria come in lots of different shapes and sizes. Some are long and skinny, most are short and fat, some are pill shaped, everything. They're all very small. Bacteria is about 1 micron to 5 microns, if that means anything to you. Most of them are visible under a microscope, especially a compound microscope. In good conditions, they can reproduce every 20 minutes. So that means if you, do the algorithmic multiplication on that, because bacteria their populations can explode in good conditions. And they're almost ubiquitous in soil the world around. It's a very dead soil, which I would call dirt, which doesn't have bacteria in it. Ironically enough, some of the soils with the least bacteria are agricultural fields and, of course, deserts. A lot of bacteria thrive in high oxygen conditions, so we call that aerobic. And then there's a whole bunch. See the ones on the far right side, the others? Those are ones that thrive in anaerobic conditions. Plants live in aerobic conditions. Anaerobic conditions in a soil will kill plants. There are plants who have learned to live in water, but they still have a workaround to get the oxygen that they need for metabolism. So if I'm looking at a microscope and in a soil sample and I see a whole lot of the anaerobic bacteria, I know something not good is going on there. It's either waterlogged soil, it's compacted so that air cannot get in, because plants need water and air and sunlight, right? Those conditions, I can tell by looking at a soil sample if the soil needs to be aerated, if it needs, that clay uncompacted, say after a housing construction, imagine how compacted soil can be. And plants can't live in that very well because the microorganisms cannot help them. Also, it's hard to get your roots through. Okay, and so let's go. Here's a nematode. Just look at okay, see all those little dots? This is nematode. They're soil microorganisms. They're basically pinworms, and there are so many nematodes. We'll talk about them in a second. But I just wanted to show you all the bacteria inside that nematode. Okay, see all those little round spots around? Most of that is bacteria, not all of it, but most of that is. And all of the little dark circles inside that nematode are bacteria. And that nematode can eat, what they say, 10,000 bacteria a day, any self-respecting nematode. So they're part of that poop loop. I mean, it's going. They're eating them and pooping them out and pooping out the nitrogen and the other things. There's also an amoeba in there up in the left-hand corner. And that brown piece at the top, that's probably just some organic matter. So soils, I think, a fascinating place. Okay. And next is, let's go, fungus, the other primary consumer. Fungus are not plants. They got in their own kingdom in the 1960s, and they inhale oxygen and exhale carbon dioxide. They are filamentous organisms, and the mushroom that grows above ground is not at all the whole organism. It's basically a fruiting body. So still not a plant, but like an apple on an apple tree. The real organism is all the filaments in the ground. And what those filaments do in the ground is weave the soil together. They make a superhighway for nutrient transfer, for water transfer, for information, and that is becoming a very interesting thing. Yeah, you might want to read about that. It's still pretty new information. And so there are different classes, different families of fungi that do different functions in the soil. One of the first is, here's how, yeah, a good explanation of how fungus work in the soil. We've got the saprotrophic fungi. See the wood fungi on the tree, and then over at the far left, that litter fungi. Saprotrophic means they eat they eat debris. They break down dead organic matter. Saprotrophic fungi do not eat live plants. So that saprotrophic wood fungi might be eating the dead heartwood, but it's not eating the live part of the plant. The saprotrophic litter fungi, the mushroom over there on the far left, is eating the leaf litter and breaking it down. And you'll see those little white strands in wood and in leaf litter. That's saprotrophic fungi. Then there's also ectomycorrhizae. There are fungi that live in partnership on the roots or in the roots of plants. And those are, okay, there's a fungal hyphae in the ground. That's at 400 total magnification, so pretty big. And you can see it's filamentous. It's like a fiber. It's got little walls in it, which I think look like bamboo, but and straight. This is a good fungus. It's dark brown. You can tell by certain markers on, is it wide? Is it got color? Is it got all these other things that you can tell a beneficial fungus from a fungus that is probably making its living off of being a parasite. Now, here is a fungus living in partnership with plant roots. Do you see? Okay, that's a little bitty seedling. Do you see the little shiny kind of yellow parts coming down? That's the plant's roots. Do you see all the big white network around it? That's a fungus. And that fungus is an ectomycorrhizal fungus, which means it lives on the outside of the plant roots. These usually partner with kind of higher succession trees like pines, some hardwoods. And look how much that increases the network for what that plant can bring in, the nutrients, the water, just the availability to the plant. So phenomenal cosmic powers. Now it's got a really big living space. And that plant is feeding it sugars, and it's getting what the plant needs. And the communication that goes on with that is still to be figured out, although it's really interesting what's come up from it. Then another one is what we call an endomycorrhizal fungi. And just to make it more confusing, they call them arbuscular mycorrhizal fungi. That started when they were first looking at it under a microscope. And do you see down in the bottom row of cells, those are plant root cells, and how those fungus spreading out, they look like under a microscope, they look a little bit like trees or maybe shrubs. And so arbuscule comes from the Latin root for tree. AM fungi, that's just what we call them. So they live outside. The fungus lives outside. They will find the plant. They will find an entry point. The plant is happy, I think, you know, if plants have emotions, to have the fungus come in because now it's living in partnership with it. And most of the inoculants that you buy to put down with your fruit trees are arbuscular mycorrhizal fungi. The trick is very specific. Each plant is specific about which kind of fungus it wants to partner with. So just getting an inoculant is not enough. You really need to at least have a wide variety or be very specific. And a lot of growers are pretty knowledgeable, or some should be, about what kind of mycorrhizal fungi, which strain you need to partner with that particular tree. So remember me talking about the madrone tree and how madrone trees are very specific. Madrones are in the ericoid family, and which is the same, the heath family, blueberries, and they need a very unusual for our area strain of of AM fungi. And so if you want to grow a madrone tree and you want it to thrive, you need that kind of fungus in the ground. The best thing that I can come up with is take some soil from around the mother tree and keep it alive, plant it with the young seedling or the younger tree and keep watering it and keep that fungus alive so that it will partner with that madrone tree. I don't know if that's the whole secret to madrones, but I know that that would at least count. Okay, so how do we know that the AM fungi has infiltrated the roots and is partnering? Well, you know, because I just spent all this money on mycorrhizal fungi and I want to know. We can tell under a microscope, and this is because they fluoresce. This is so cool. I bought a special unit for my microscope that when I turn it on and it focuses on AM fungi inside the cell roots, they fluoresce. Have you ever taken a UV flashlight and found scorpions or mushrooms? It's similar, not the same thing, but similar. They'll usually see this big flash of green. I mean, it's not a flash, a big constant. And you can tell how well that inoculant worked by looking at it under a microscope, which is handy. And now there's another category. So we went through saprotrophic fungi, which eat dead stuff, mycorrhizal fungi, which we've got the ecto and the endo, the AM, which help the plant roots. And now they're discovering endophytic fungi, which live their entire life inside a plant. We never see any results of them outside except for maybe pollen going through the air. But they partner with specific plants, particularly grasses have a lot of endophytic things in them. And sometimes, you know, when grasses will freeze and become toxic to livestock, it's looking like it's not the grass itself, it's that endophytic fungi. That their fungi are super smart. I mean, they can do all kinds of things and they can change from being a big help in a plant to becoming a pathogen, either to the plant or to a plant's predator, being in this case livestock. So endophytes live their life. In fact, they are transferred inside seeds, which it makes me wonder about, as I'm trying to grow plants and scarifying seeds, particularly acid scarification of seeds. Is that okay? Am I killing the endophytic fungi in the seeds or on the outside of the seeds? Of questions, lots of questions. And there are answers, but it takes some research. And of course, there are, we've talked about beneficial fungi, and as we all know, there are some fungi that are not beneficial to plants. And they're usually lower, more primitive forms of fungi that are less complex. This is, and I can't even say the name, Pseudoperonospora. It's downy mildew. That's what downy mildew looks like under a microscope. So if I were looking at a soil sample and I see a whole bunch of downy mildew, I would go, ooh, oh, not good, not good. And you can tell that it's a light color. It's real irregular in its thickness. There are little telltale markers like that about fungus that might be pathogens. Here's a photo of, this is Fusarium wilt in the soil. That's a fungus. And I think those are the spores of it actually. But yeah, you can see all these things in soil. So one thing I want to leave you with about fungi is that it is a tapestry underground. It is woven. These are filaments. And that is why digging in soil, why tilling soil is destructive to the plant and to the soil food web, because for the main part, the worst thing is you're tearing up all the fungal networks, and the fungi take a long time to recover. If you do it once, they're going to recover. It'll take a little bit to rebound. But if it happens repeatedly, fungus can't handle that, and they will suffer. They will suffer, and so will the plants. Okay, let's talk now about these other guys. The protozoas and the nematodes are what I'm going to particularly talk about. Springtails and mites, which you can see in there, are things you can actually see with your bare eyes, but protozoa and nematodes are much smaller. So protozoa are one-celled creatures. I think you might remember them from biology in high school. There's three kinds of protozoa. They're all one-celled, and they eat bacteria for the main part. Sometimes they eat each other, which is how this is a web, not a exact chain. Flagella have a tail or a flagellum, one or two or three, kind of depends. And they just push themselves or sometimes pull themselves through the soil with this whip-like little protrusion. Flagellates move slowly, and they kind of bumble around in the soil. And flagellates are very aerobic. They live in aerobic conditions. So we're glad when we see flagellates in the soil, because that indicates that soil's aerobic. On the other hand, cilia, the bottom one, see all the little hairs around the outline there? Lots of hairs, cilia, as they call it, and those are ciliates. Move super fast. They're like Roman rowers in those long skull things. They can shoot through the soil. And if I just see something whiz past and I wasn't quite sure what it was a ciliate. But they live in either anaerobic conditions or what's right between, a soil that's going anaerobic. You see a lot of them in pond water, for example. So when I see ciliates in soil, I begin to go, okay, I don't want to see too many of those, because that indicates that soil's going anaerobic. And that is a picture of a flagellate. That's about the best I can come up with. They're little bitty. Can you see it? Can you see those two little flat flagella coming out of it? And it's just, bumping along, kind of looking for bacteria to eat. And there is in the center, and it's hard to see because, again, they're kind of transparent. Can you see that? I wish I had a pointer. Can you see that ciliate? It's got lots of little hairs, cilia coming out from it. And sometimes they slow down and they'll get in. Okay, see that big bunch of brown stuff over on the left-hand side? That's organic matter. That is, in fact, some probably humus. And they'll get in there and just work it and go around to find the bacteria that they like. Both ciliates and flagellates are pretty particular about the kind of bacteria they particularly want. I don't know what that is, but they know, and they'll just dig around in all kinds of stuff till they find it. And then ciliates will go whizzing away at light speed. And okay, now let's talk about amoebas, and they are the other protozoa that affect soil biology. We all remember the chaos amoebas, the oozing amoebas that you've seen in biology class. I don't have a picture of one of those. I'm sorry. I wish I had a video of it because they're fascinating to watch. In fact, it's just mesmerizing. But there are other kinds of amoebas. These are, see that those are two hooked together. They look like little angel wings on the beach. That's a testate amoeba, and the testate means they have a shell. And they come in a lot of different forms, but this is a pretty typical form. And what it's doing is dividing and making a new one. The one that has all the stuff inside of it, that's the mother amoeba. And the new one that's empty inside is that first one is making the test, the shell, and then it's finally they will separate and it will go on its way and start hoovering up bacteria too. And you can see the original amoeba, the one on the left, has all that bacteria in it. They're bacteria vacuuming up machines, and then they spit out what they can't eat, and here we go, round the loop. There we go. Okay, that is another kind of, that's two kinds of testate amoebas. That one on the right-hand side, that's the regular testate, and the round one is called an arcella, and I think they're pretty. They look like a donut if you see them in a profile. It's got that dark thing in the center is kind of a caved, hollowed out curve in the thing. So arcellas and just regular testate amoebas. And this is a nematode, which is, nematodes are on the next level. They consume bacteria. They also consume protozoa. And nematodes are in fact animals. We're out of the world of just protozoa now and into full animals. In fact, nematodes are the most numerous animal in the world. And we've all got nematodes everywhere. They say if you took away all of a person's DNA, just vanished everything that had their DNA, you would still be able to see an outline of them just from the nematodes left, which I don't really like to think about, but you know. Okay, and this, most interestingly, is a female nematode. You can see about two-thirds of the way to the left there, that big egg inside her. See that? That's a pregnant nematode, which I bet you've never seen before. I want to bet. And the tail is on the left. The head, the mouth is, they're only just a mouth. There is no head. It's all mouth, is on the right-hand side. And this one, I can tell from the shape of the mouth and the shape of the esophagus, is a bacterial eating nematode. They have different functions. There's bacterial eating. There's nematodes that eat fungus. There are nematodes that eat plants, which you have probably experienced. And so most nematodes eat other nematodes. So that's a thing. And come on, here's a nematode crammed full of bacteria. Isn't it pretty? I think nematodes, and they thrash. You just can't even see them. They're moving so fast. And then the other thing I find in soil samples is plastic. So much plastic. I'm always disappointed at how much plastic. This, I believe, is blue. And so to me, that would mean it's from one of the blue tarps that I will never buy again. But the soil is full of microplastics, and you can see them, and they're shiny, and they're there. They're going to be there for a long time. Okay, so let me skip to this next slide. And then there's other things below ground, which you're familiar with, earthworms, moles, spiders, which are visible to us, so we're more familiar with those. And they're part of the soil food web too. So here we are at our succession again, and knowing that what's going on underground is every bit as important as what's going on above ground. And when we start with just weedy annuals, we have mainly bacteria in the soil. And as we move through succession, we get a different, we get more, we have, okay, so there's bacteria and fungus. We start off with bacteria. That's a basic soil. So say fungal to bacterial ratio here, there would be one piece of fungus for every 10 bacteria. So it's highly bacterial dominant. And as we go along, we'll see that more fungus live as the soil gets more complex, as the community, the underground community gets more developed and the fungus has time to grow. And so now when we get to grasses and perennials, we have one fungus for every 2 bacteria. So more fungus in relation. If you are growing a garden, this is where you want to be, somewhere between this and the grasses, shrubs, perennials, and that 1-to-1. So you know you need to suit the plant that you want to grow, create the soil that they want to live in. And a fungal to bacterial ratio is real important. This 1-to-1 fungal to bacterial is perfect for a lot of kinds of trees, for many of our soil, for many of our garden vegetables. If you want to grow brassicas, you need to go back because brassicas, the kale family and the broccoli family are pioneer plants, and they want almost all bacteria and no fungus. They are one of the few plants that does not partner with fungus, and they don't care about fungus. And then as you go along, then in a climax forest, you get maybe 10-to-1 fungal to bacterial. So now you're more heavily fungus, all the way in an old growth forest to like 1,000 fungus for every 1 bacteria. But that would not be good soil to try to have in your garden, because that's not what mid-succession plants, like what we eat, want. So gauging where you need to be on this scale to grow the plants you want to grow is where you want to be. And how we do that is, how you protect soil is through, well, let's take the principles of regenerative agriculture. You don't have to know all the soil science. You don't have to be able to look under a microscope and pick out how many bacteria and how many fungus, but know what does work and what creates what you want. Minimize the soil disturbance, because then you're not killing the fungus. You're also not killing nematodes or any other of the things. Let them down there, let them do their work. Maintain a soil cover. Keep it covered to keep the moisture and the plant and the microbes protected. Also give them food. They love to eat mulch. Diversify crops in this case, but in your case, it might be native plants. They need all different kinds of plants, feeding all different kinds of exudates to meet the needs of all of the soil food web community. Keep roots in the ground. If the soil doesn't have plants to feed off of, it's going to eat off itself. And integrate livestock is the last one, and that may seem kind of out of context with native plants, but all of our native plants evolved to work with livestock, and they're part of the system. And if you take the livestock off, well, now you've got to do their job, which is okay. You can mow, you can, specifically, not too much, intelligently. You can do some of the work that the livestock was doing. Okay, and that's a lot I know it's really barely scratching the surface on most of this, but I wanted to give you an introduction and let you know that you can find so much more information. There's a really good book. Let me get it. Here's a book I can recommend. It's Teeming, Teaming, with Microbes. Covers a whole lot of what I've talked about today. And it's by Jeff Lowenfels, and it's part of a series that he wrote. And there's teeming with teeming with nutrients, teeming with fungi, teeming with bacteria. Anyway, it's a great series. And even just this one book is real informative. Okay, and so do I have questions? Jeff, Jeff, Lowenfels, Lowenfels. How are we doing on time over there? We okay? Yeah. Any more questions? Yeah, right at the back there. This one. Mm-hmm.
I recently restored an older home that had been rented for 10 years.
Can you hold that closer? I'm having a little trouble.
I recently restored an older home that had been rented for 10 years. And to say my yard is under abiotic stress is an understatement. I have scheduled core aeration and composting, but we have to wait until the temperatures drop for the compost. Is there any other suggestions? I plan to do this for at least a period of every 6 months for a couple of years until we see what's going on. As far as quality of compost or any other tips you might give me in order to speed up this restoration, because I'm to the point of almost no plants left with the drought and the lack of care and no supplemental irrigation.
One thing to know about microorganisms is when they're seeing conditions that don't meet their needs or that they think are endangering their lives, they can insist or they go dormant or they make spores. And so they will be there in the soil, but inactive. Now, if something happens dramatically, like a flood or something, it can catch them off guard and they won't have time to do that. But if it's a slow thing, like a growing drought, then they have insisted or encapsulated, and the spores and the are going to be there to hatch out later when conditions are right. So take heart in that. About compost, oh wow, that's a whole talk. What is compost? There is no definition of it, and what most people call compost is what Elaine calls putrefied sewage. The refuse industry in the 90s figured out that they could, let this stuff sit, rot down, and then sell it instead of having to landfill it. And so they call it compost. It ain't compost. Good compost has an entire soil food web active in it. It's just lacking the plant roots, and it needs to be in there. Good compost should not be hot. That's not made compost. That is not finished compost. Good compost should sit there long enough to have the fungus begin to regrow in there. You should have short pieces of fungus. So it's hard to find a good compost anywhere, but particularly I think in this part of Texas, I haven't found much, but I haven't done an exhaustive search, and I would be really happy to find out about some. Compost can be like sourdough starter. You just need a little bit and then put it in a good situation, and those microbes will multiply and grow into the good material you've given them. So that's something. As far as your situation with aeration, does water soak into that soil very well or just sit there? You're going to have to wait for rain then. But they make tools that will aerate. It's not tilling. You're not turning soil over, but you're putting holes in it. You can either rip them or they make there's a broad fork. They're not cheap, but they sure are good tools. And there, you stick them down and move them. What you're doing is just opening up air pockets. So, good luck with all that. The best to you. Did I see some other questions? Yes, with the hat.
Yes, that was so exciting. Thank you so much, your talk. When you spoke of the bacteria, I guess when it dies, it provides nitrogen to the soil. And
I think I'm hard of hearing. I can't,
Oh.
barely getting it. Uh-huh.
okay. You had mentioned bacteria provides nitrogen to the soil and that it gets locked up and doesn't wash away when there's floods and whatnot. What is a mechanism that keeps it locked up in the soil so it doesn't wash away?
I'm glad you asked that. Bacteria make glues. Fungus does too, but bacteria are really good at it, and they can glue themselves to things. You know this from your own teeth. You get that plaque that gets, and if you don't brush your teeth, you forget, and you can feel it. That's bacterial glues on your teeth. And so they stick to things, and they can keep themselves from being dissolved and washed away. There's some other interesting parts about how bacteria, so a compost extract is just where you put compost in water, get it more or less dissolved and then pour it on. And that's just water with compost in it. But a compost tea, which is good, it's good stuff, but a compost tea, you brew it and the bacteria over 24 hours or 48 hours has time and it has food to develop these glues. And then if you spray it on plants as a leaf spray, then the bacteria have those glues and they will stick to the leaves and the stems. And so it works really well. If you're going to do spraying, you might want to do a compost tea rather than a compost extract. What would she say?
Where does biochar?
Biochar, it's like high-rise, high-density housing for especially bacteria, but all kinds of microbes, which is why you should charge the biochar before you put it down, because it's the burned out carbon. Is all that's left is the cellular structure of usually wood. And so it's really like a high-rise apartment house. But if you just put it in without charging it, all of the bacteria and the microbes are going to leave your situation and go be populating it. And so you have, it looks like you've had a population dive, but what's actually happening is they're just setting up housekeeping. But and then once it's full, then they'll be back in your soil. But if you go ahead and charge it, that is, get it all full of microbes first and then put it down, you don't see that initial dip. How do you do that? You soak it in compost tea. You just put it in your compost pile. Just leave it in contact with microbes and in good soil for long enough that they have time to set up housekeeping. It has other benefits too, but that's the one with the microbes. Mm-hmm.